Good morning to everyone. Today I am here with a very interesting subject, plant breeding. So plant breeding is the art, science and technology. Plant breeding is a art, science and technology. When we using this art, science and technology to change the genetic architecture of a plant to get the maximum benefit by the farmers or users is called plant breeding. There are lots of scientists, lots of authors define the plant breeding in different terms. According to the Simmons, plant breeding is the current phase of crop evolution. According to the Vavilov, N.I. Vavilov, plant breeding defined as a crop evolution directed by human. Crop evolution directed by human, according to the Vavilov. Vavilov is a very famous scientist because of he gave the concept of center of origin. He gave the 8 center of origin. Because every crop have the different kinds of the center of origin, like rice. The center of origin of rice is India, Southeast Asia. The center of origin of wheat is Southwest Asia. The center of origin of soybean is China. The center of origin for cowpea is India. The center of origin for groundnut is Brazil. So different plants have their different center of origin. So today I am here with some introduction about the plant breeding. So plant breeding is the genetic adjustment of plant for the social, cultural, economical and technological aspects for the environment. Plant breeding is a science, art and technology. And when we using the art, science and technology to improve the different characters of the plant in such a way to get the maximum benefit from that plant. So this is all called the plant breeding. So why we calling the plant breeding is a art? Because in plant breeding we have to be develop the some important kinds of variety. And that important kinds of variety is developed by selection. And during the selection procedure, the human or scientist can adopt some imagination, some art techniques on the basis of observations. So we call this is a art. Next, the plant breeding is a science. Why we calling the plant breeding is a science? Because to developing the new variety, to producing the new variety, we have to be follow a systematic procedure. Some theoretical concepts, some procedure, some protocols are also followed to developing the variety. Like the variety is developed by the different plant breeding methods, such as backcross breeding method, pedigree method, bulk breeding method, pure line selection, mass selection, recurrent selection. So these are the breeding methods, and that breeding methods have the different kinds of procedure, different kinds of steps. When we follow the different kinds of steps, then the last product is our variety. So this is the systematic procedure. So we call the plant breeding is a science. Next, the plant breeding is a technology. Plant breeding is also a technology because with help of plant breeding we making some useful product, and that useful product is our variety, our hybrid, and that hybrid and variety is cultivated in a large scale by our farmers. So this is the concept of the plant breeding. So plant breeding is changing the genetic material, changing the DNA of the plant in such a way to improve the genetic character of the plant. So in case of plant breeding, there are objectives. We have to decide the objectives of the plant breeding. but the objective of plant breeding is decided on the basis of the country. The developed country, the breeding objective of the developed country is different, the breeding objective of the developing country is different. India is a developing country, USA is a developed country. So in case of USA, the first breeding objective of the plant breeding in USA is quality, not yield. But in case of India, as we know that India is a developing country, so first priority in our country to maximize the yield potential firstly, next quality. Because first of all the human being in our country is get the maximum amount of yield, because quality is the next objective. First of all they could got the benefit, and next objective is the quality. But in case of developed country, the first breeding objective is to quality, next yield. So today I am talking about our country related breeding objectives of the crop. The first breeding objective is the higher yield, and this yield is achieved by to taking the some different types of indirect character of the plant, because yield is a quantitative character, yield is not a qualitative character. Because in case of quantitative character, that quantitative character is controlled by polygenes. As we know that the every character of a plant is controlled by gene, and gene is a segment of DNA that produce a functional product, that produce the protein. This is called the gene. So in case of to improving the yield of the plant, we have to be focused on the polygenes, and that polygenes means more than 2 genes, more than 3 genes, and these polygenes are situated in the different kinds of the chromosome, not present on the same number of chromosome. So that polygenes is present on the different number of the chromosome, because every individual, every living organism have the different kinds of the chromosome number. In case of rice, their chromosome number is 2n equals to 24. In case of maize, the chromosome number is 2n equals to 20. In case of pea, Pisum sativum, the chromosome number is 2n equals to 14. In case of barley, Hordeum vulgare, the chromosome number is 2n equals to 14. In case of bajra, pearl millet, the chromosome number is 2n equals to 14. In case of soybean, glycine max is the botanical name of soybean, the chromosome number is 2n equals to 40. In case of wheat, Triticum aestivum, the 87% area in our country is occupied by the species Triticum aestivum, 2n equals to 42. So the different species have the different kinds of the chromosome number. So I have today the talking about the breeding objectives of the plant, breeding objectives of the plant breeding. So the first breeding objective is to yield, to maximize the yield, and yield is maximized, yield is increased with help of, we have to be, or scientists focus on the different kinds of the indirect characters, because yield is a quantitative character, and quantitative character is controlled by the polygenes. Next breeding objective is the quality. Quality is also important, because you have to be increase the yield of the plant, rice or wheat, but the quality, the protein content is very less, the vitamin content is very less in those kinds of variety. So the farmer cannot cultivate your new variety, so ultimately your variety is flawed. Because of that, the quality is also other parameters of the plant breeding to improve the quality in terms of protein content, in terms of the vitamin content, in terms of the mineral content, in terms of the carbohydrate. These are the important points related to the quality. Next breeding objective is to develop a variety resistant to disease and insect pest. Because of that, the maximum yield of a variety or plant is reduced due to the infestation, due to the infection of the disease as well as insect pest. So ultimately your variety is high yielding, good quality content, but the lots of infectious agent is surviving in that plant, lots of pathogens are infected your plants, so your yield is drastically reduced. So how to be control your yield potential? So this is also a breeding objectives of the plant breeding, resistant to disease and insect pest. We have to be produce such kind of variety, that variety is resistant. Resistant variety means this variety or this hybrid cannot infected by any kinds of the pathogens, cannot infected by the bacteria, cannot infected by the fungus, cannot infected by the any insect pest. This is the also breeding objective of the plant breeding. Next breeding objective is to tolerance against abiotic stress, because nowadays the climate change is a major issue. The environmental condition, the atmospheric condition during the kharif season, during the rainy season, no much amount of precipitation is observed. In case of rainy season, drought condition is observed. So due to that reason, we have to be develop a such kind of variety, that variety has the potential to survive in those conditions, in drought condition, in salinity condition, in acidic condition. So this kinds is called abiotic stress. Abiotic stress means non-living things. The plant face the stress by non-living thing, like temperature, like heat, like drought, like salinity, acidity. These are the important parameters of the abiotic stresses. So this is also a breeding objective in plant breeding, to develop a such kind of variety, that variety has the potential, that variety has the capacity to face this type of situation, abiotic stresses. In drought condition, our plant has the potential to increase the higher yield. The next breeding objective is to develop the early maturing variety, because of the climate change, the drought condition. So our focus is to develop a such kind of variety, their maturity duration is very less, short duration variety. Suppose in case of rice, there are lots of variety, and lots of variety have the different kinds of their maturity duration. I have to giving an example, the BPT 5204, Sambha Masuri. The duration for the maturity of the Sambha Masuri is 145 to 150 days. 150 days. But also a other kind of variety of the same crop, rice, they mature in 90 days, 80 days, 100 days. IR64, IR36, IR64 DRT1, Birsa Vikas Dhan 109, Birsa Vikas Dhan 110. These are the variety that mature in very early time, short duration variety. So the merit of short duration variety is to easily complete their life cycle in short period of the time. So this is the also breeding objective in the plant breeding. We have to be developed in such kind of variety, their duration is less. The other breeding objective is synchronous type of maturity. What is the meaning of synchronous type of maturity? Synchronous types of maturity means the every pods, every grain mature at a same time. You have to be seen in the field. In case of moong, green gram, Vigna radiata. Vigna radiata is the botanical name of green gram. There are several, during the time of harvesting, there are several picking is required in case of moong. So the labor cost is increased. Lots of times are spent during the harvesting of the green gram, because the different pods have the different kinds of harvesting period, different kinds of maturity duration, not mature at the same time of all the pod of the same plant. So this is also the breeding objective of the plant breeding, to every pods, every grain mature at the same time. When every pods mature at the same time, so the major benefit is the cost of cultivation is less, because at a single day the laborers can harvest all the crops, because mature the crop in the same time. So this type of breeding objective is followed in case of green gram, pulse crop, black gram. The other breeding objective is also we have to be develop the non-scattering type of variety. We have to be develop the non-scattering type of variety. What is the meaning of non-scattering type of variety? Scattering means during the I have to be given the example of the same crop, green gram, moong. During the maturity period of the green gram, a small touch by human in the pod, their pod is open, and during the harvesting the lots of grains are scattered in the field. The maximum loss is taken during the time of harvesting of the green gram. So we have to be develop such kind of variety, that variety have the not scattering problem, not open the pod during the time of harvesting. So this is the meaning of the non-scattering type of variety. During the time of harvesting, after the small touch in the pod of the green gram, their pod is not open. Okay. The variety is developed according to the breeding objectives. First of all, we have to decide the breeding objective, and after the decide of the breeding objective, we have to be adopted. We have to be taken some breeding method, because there are lots of breeding methods is available, but the different kinds of breeding methods is adopted according to the different kinds of the plant. Different kinds of the plant means the breeding method is adopted according to the nature of pollination of the plant. Means the self pollinated crop, cross pollinated crop, often cross pollinated crop. The self pollinated crop have the different kinds of the breeding method followed to developing the variety. When we making a new variety in cross pollinated crop like maize, we have to be adopt some different kinds of the breeding methods. So this breeding methods is dependent on the nature of pollination of the plant. The self pollinated crop. Self pollinated crop means the transfer of pollen grain from anther to stigma within the same flower of the same plant. The flower is hermaphrodite flower. Hermaphrodite flower means the male part or female part, the stamen and pistil present in the same flower. So in the same flower, the stamen and pistil is present, the maximum chances is for self pollination, autogamy. Autogamy is synonym of self pollination. So in case of self pollination, we have to follow the pure line selection. Pure line selection method is only adopted in the self pollinated crop as well as often cross pollinated crop. But this pure line selection is not used in case of the cross pollinated crop like wheat, like maize. Wheat is a self pollinated crop. Rice is the self-pollinated crop, but maize is the cross-pollinated crop. Papaya is the cross-pollinated crop. Cross-pollination means transfer of pollen grain from anther to a stigma of the different plants. Pollination between different plants is called cross-pollination or allogamy. I have to be mentioned here the other type of pollination is the geitonogamy. What is the meaning of geitonogamy? Autogamy, allogamy and geitonogamy. Autogamy means transfer of pollen grain from anther to a stigma within the same flower of the same plant. In case of allogamy, allo means different. Transfer of pollen grain from anther to a stigma between the different plants. Plants is different in case of allogamy, but in case of geitonogamy, plant is same but flower is different. The transfer of pollen grain from anther of the one flower to the stigma of the different flower of the same plant. Here plant is different, but pollination between different flower. Pollination between different flower of the same plant is called geitonogamy, and this condition is found in the case of maize. Geitonogamy condition. So this is all about the introductory part of the plant breeding, and I have also discussed some important kinds of the breeding objective. In the next lecture I have to be prepared for the activities of plant breeding, which kind of activities is conducted in case of plant breeding, which kind of works is adopted in plant breeding, and in the next lecture I have to be discuss a plant breeder have the knowledge about the different kinds of the subject, different kinds of discipline in the next lecture. So this is all about the plant breeding. Plant breeding is a subject, is a discipline, is a art, science and technology. When we using this art, science and technology to improve the genetic character of the plant, because to maximize the yield, after the maximizing the yield, the farmer got the maximum benefit from that. They improve the quality content. All the previous character has been improved with the help of plant breeding. All the genetic character means height of the plant, grain quality of the plant, number of branches of the plant, number of grain per pod of the plant, vitamin content of the plant. All these things are focused in case of plant breeding. In plant breeding we have to improve the plant, improve the variety, and that variety have the different kinds of the feature from the previous kind of variety, previous traditional types of variety, and this variety is developed or produced by the respective plant breeder. Plant breeder produce the variety. So this is all about the plant breeding, and there are different kinds of breeding objectives. The first breeding objective is higher yield, next quality, then resistant against disease and insect pest, next tolerance against the abiotic stresses like the temperature, like the heat, like the flood, like the drought, salinity, alkalinity. Other breeding objective is to develop the short duration variety, the synchronous type of variety, the non-shattering type of variety. These are the breeding objectives. These are the important types of the breeding objectives. The other breeding objective I have to also mention, to reduce the toxin level of the plant, because the lots of plant have the toxin. In case of Lathyrus odoratus, commonly known as sweet pea, khesari, in the crop sweet pea, the BOA content is here. The BOA is a toxin compound, BOAA, BOA content, and due to that toxin, the human can be paralyzed after the taking so much amount of this BOA content during the feeding of the Lathyrus odoratus. So we have to be develop the such kind of variety. That variety have the very less amount of the BOA content. This is also breeding objective. In case of mustard, here the toxin is erucic acid. Erucic acid is the toxin in case of mustard, brassica. So this is also the breeding objective. Develop the variety in less amount of the toxins. So thank you.